Gotta do it. I'll leave a little bit of time toward the end of the time period here if you have any lingering questions. Also, a reminder, between now and Friday, I will be checking my email into the evening regularly in case if you just have, a quick question that you want answered on the fly. I will try to respond quickly. Okay. So far, the exam is still in draft form, but it's starting to take shape. And it seems like there's more points coming from plant cells and tissues and the plant body, which is root, stems, leaves, in terms of the multiple choice portion. Okay, so far that's a little bit heavier weight than what I got going on with the fungi, algae, bryophytes, and seedless. In the sense that, if you look at each group, each topic, so far there's more points coming from the plant body and plant cell and tissue. If that changes dramatically, I'll let you know, but that will help you focus a little bit more, make sure that you're not neglecting the plant body and cells and tissues. Okay. Essay portion, I have not gotten that firmed up yet, but what I'm leaning toward doing is, instead of like one big essay or something like that, doing several short answer. And at least some of the questions, I give you options so that it's easier to answer, I guess, in the sense of, if I give you some options, hopefully one of the options will go, oh yeah, I know what to say. Okay. So that's at least one or two of those questions would probably be like that. And so when you're studying for essay, it's often any kind of process explanation or something significant that we talked about that you think, designing a good multiple choice question on process questions is challenging. And so that's why essays or short answer get used for that. So multiple choice can be effective, but not for some of the more something that you need several sentences to really flesh out and explain. Does that kind of make sense? What I'm saying? All right. So, and then two, remember this is an applied plant science class. So, we do have sections that we've been talking about with each group of some of their applied uses, right? So don't neglect looking at those ideas as well, because this is one of the important aspects of this class is how do you use this information and then apply it in the real world. Okay. So by the way, I think most of you know I enjoy fungi. Have you seen this? It's at the store in the basement of the library, Sundries, I think it's called. Odyssey Sparkling Mushroom Elixir. I didn't know that they had such a thing. This is dragon fruit lemonade, but they make a couple of other flavors. And this one, when I'm like, okay, what's going on with the fungi here? They have lion's mane in here and cordyceps. And then the energy idea is some caffeine too, but lion's mane and cordyceps extracts, I'm guessing. So, yeah, this is dragon fruit lemonade, and I think they make blueberry lemonade or something. So Odyssey, I think it was like $3.80. So it's not cheap, but I thought, oh my gosh, they're carrying something that's fungal based. It's like, that's pretty cool. So I had to try it. So it's, it doesn't taste like mushrooms. If you're wondering, you can't taste it. It's just the flavor that they have added in, the dragon fruit in this case. Okay. So we left off looking at gymnosperms, and I don't think you had enough lily flowers in lab. Did you? Did you have lily flowers to look at yesterday? I don't think you did. So you're probably going to get that again next week. That was a mistake when the local shopping got done, it didn't get picked up on. So we will redo that for next week. But flowers are not part of the gymnosperm life, but we do need to think about the cones as a really important part. So this is a comparison slide to the previous one in the slide set, looking at the idea that in the other groups that we've covered, bryophytes and seedless vascular plants, it's the spores that are released out. They have to fall somewhere suitable and then germinate and grow into the gametophyte. And then the gametophyte has to go about its job of making egg and sperm. So it's, you know, those spores are kind of just flung away from the mother plant, so to speak, and it's like you're on your own. With this idea, now we have spores being retained, and so they're protected by protective tissue. The gametophyte will get produced from that spore growing, but again, it's all retained. It's held on the sporophyte and protected. And so this protective mechanism and not releasing it like that, like they do what we just saw with the spores being released, they think that has really helped in the survival of this group of plants. And then when you look at the seed, which is the actual thing that gets released, the seed is much more well designed to handle long periods of having to go dormant because conditions aren't good, whether it be too cold, too hot, not enough moisture. And so most seeds, the embryo is inside there, but so is some dose of nutrition that comes with it. And then you have that really tough seed coat. So some big differences when you compare the gymnosperm to the other two groups we've already looked at, okay? And so when you're out hiking around, it's the sporophyte that you are seeing, is that portion of the life cycle. And so what I'd like to, so this slide is pretty much summarizing what I just said, that the spores stay on the sporophyte. Now, what we're going to find in the gymnosperms and the angiosperms is that, remember, there's megaspores and microspores now in these two groups of plants. And what happens is the megaspore doesn't get released. It just stays in the sporangium tissue, and so they call it megasporangium. And so everything stays in there. And then what happens is, and then with microspores too. So the microspores, they don't get released. They stay in the microsporangium tissue, and they will develop into, well, eventually the pollen grains, okay? So what we also see is no free water is necessary, and that's another big advantage, because now the plants don't have to rely on moisture in the environment to complete their life cycle. So when the gametophytes do end up getting developed, they are in, tissue of the sporophyte. They're all protected by sporophyte tissue. So they're not independent anymore. They're dependent on the sporophyte. And it's the seed, not the spores. So this is a definite shift. So a couple of key things here I'd like you to think about. Here they're looking at zooming in on the female cone and looking at the scale. So if you take one of those scales off the cone and look at it under the microscope, you would see the megasporangium tissue. Remember, sporangium tissue holds spores. In this case, it's the female spore. And then that female spore doesn't get released, it stays. And then that has to go through, cell division, and grow to become the gametophyte, right? So instead of being released out, if we go back to this picture where you see these spores, get released out and then they germinate and grow into the gametophytes, right? Well, with the gymnosperm idea, whoops, not that one. With the gymnosperm idea, you have the spores retained, but they still do develop into a gametophyte. In this case, because it's the female gametophyte or megaгаметophyte of the female cone, right? And so the gametophyte's job is to produce eggs. So you see that happening. And then the male gametophyte or the micro gametophyte is the pollen grain. Technically, that's a good way to think about it. The pollen grain is really the male gametophyte, and then that will eventually the sperm will get released and fertilize the egg. And then you have the development of the zygote and then the embryo. So when the zygote keeps growing, eventually then we start shifting and calling it an embryo. Right? And then you see that there's food supply. Okay, actually it's the female gametophyte will serve as food supply. So it's going to nourish that baby plant once it starts to germinate and grow. The baby plant can't do enough of its own photosynthesis, so the tissue here will get, the nutrients will get siphoned into the embryo and used. Okay, so things are retained. The gametophyte develops on the sporophyte. So this is the overall life cycle, and I'm not going to go into the details of this in terms of what I'm expecting you to know, other than the basic idea of the alternation of generations and the zooming in on the female cone and the male cone, the idea that you get, that ovule is the region of the female cone that we need to zoom into. And inside the ovule is where you're going to have all that development we just talked about, where the megaspore will develop into the gametophyte, which will produce an egg, and then it's ready to go, ready to be fertilized. And then the pollen grains also are going to develop into the male gametophyte. And when you have pollination in the gymnosperms, it's delivery of the pollen to the ovule. And eventually sperm are going to show up and fertilize the egg. There's usually 2 sperm, by the way, that get released in the gymnosperms. There'll be 2 sperm per pollen grain, just 2. And then it's a race to the finish. 1 sperm can fertilize the egg, the other one loses out and dies. Okay, so it's just a 50-50 shot which sperm is going to be the winner on that. All right, so 1 sperm fertilizes the egg, the other one dies off. We're going to see in the angiosperms, that's a very different story. Both sperm win. They each do something important. Okay. So once you have the fertilized egg, the zygote, and then the embryo, and then eventually the full seed development. And it's the seed that gets released. So I'm not going to ask you for the details of this life cycle, not in the gymnosperms. In the angiosperms, we'll get more into it. So it's really, the cone tissue where you got a lot of key things happening. And so when they look at that and they're trying to figure out what are the cones, they really are a type of strobili. What we saw, similar to what we saw with the bryophytes, and looking at the idea that you have sporangium tissue that's closely packed together, and that a cone does fit that definition. And they say it's composed of branching tissue, not leaf tissue, and that there's a range of size. You can have the juniper berries, so to speak, but those are really cone tissue. That's cone. They're cones. They're not berries. And then cycads are being shown down low here. Cycads are the only, as far as I know, they're the only exception. Normally, they say with the gymnosperms, normally what happens is the female cone is always bigger than the male. The cycads are a noted exception to that. And so what you're seeing here is on the left is the male cone, and it is bigger than the female cone. But if you put them off to the side, all the other gymnosperms, the female cone is bigger. And so the pollen cones are smaller. And then pine cones, the mature pine cones, they will get woody at maturity, the female pine cone. And so when you're looking at pine cones, I'm sure you've seen this through your life already. Sometimes the scales are wide open, sometimes they're partially open, sometimes they're all the way closed and sealed up. And so that is depending on their life cycle, where they're at in their life cycle. And so with the pollen cones, of course, the job is to release the pollen grain. And then hopefully some of those pollen grains will waft over, usually through wind currents that we're thinking about here, will go to the ovule of the female cone. And if that happens, so when the cone scales are widely separated like that, that means a couple of options you got going on there. It could be that the ovules are like, all right, let's get pollinated, and pollination is needing to happen. But the other reason that they also will open up their scales like that is if they're ready to release seed. So it could be either one of those cases why the scales are open. So anytime you see an open pine cone, look on the top of the scale and see if you can notice any seeds or not. And they might have fallen out or fluttered out of there. Look for that. Male cones are fairly simple in structure compared to a female cone. And so you're just seeing that here you have, this picture was taken before the pollen was fully mature. So these are all microspores. So each one of those regions is the microsporangium. All right, so if you just look at, oh, stop it. So that circle I just made, that's a microsporangium. And then inside are microspores. And then eventually those microspores will become the pollen grains. They're just not there yet. They're immature. But pretty simple in design. And then here you're seeing on the left, the three options. The one on the bottom is fully open. The scales are fully open. The one off to the left is like partial. And then the one to the right is all closed up. And so if they're all closed up, chances are, hopefully, that they got pollinated. The ovule received some pollen. And once that happens, the scales start to close up and actually fuse to the central stem of the cone. And then they do that because they got a long process now. It takes a year and a half for everything else to happen now. So pollination happens, but then getting everything else to happen in terms of getting the sperm to the egg, that takes a while, and then getting the embryo to develop and the seed to mature. A year and a half. So that's a long time. So when you know that, it does really kind of make sense that they close up and protect that whole process. On the right here, I thought this was interesting when I was looking at cone design. Several sites were saying you can use them as a way to assess humidity or possibly a rain event in the near future. They say that the cones can open and close based on moisture levels too. So just be aware of that. That's you'll see that happen. I think people usually do that with cones that have already shed their seed. They'll keep the cones and see if they can get it to do that. And then I showed you this last time, how you can have pine cones on one tree and on one branch even at different stages of growth, where obviously the newest pine cone isn't ready to do much of anything in terms of sexual reproduction. But this one here, this 1 year, this one has, hopefully all those ovules have been pollinated, and so they've sealed up. And then this one here is, after about a year and a half, the scales open up and the seeds get released. And then don't forget that there are some species, not very many in the grand scheme, but there are some species that need fire in order for their cones to open. They won't just open up their scales when the seeds are ready. And that's a strategy based on the adaptations they have to the ecosystem. They need to have kind of a clear-cut area, fire burning down all the competitors for them to successfully germinate and grow and gain a foothold. So those are pretty unusual examples. So when you look at a scale of a cone, they each have, if everything goes okay, they each have two ovules there. And they're all the scales, if you look down on it from the top, they're arranged spirally on the central stem. And like I said, in the spring, when pollination is happening, they're going to have their scales open in order to get the ovules pollinated and then they close together once pollination occurs. Here's like the top half of a pine cone, female cone. And so you can see each of the scales here, and you got to kind of zoom in. You can kind of see on some of them anyways, like a little blobby visual blob toward the top of the scale. These two in particular are pretty decent. Oh, that looks pretty good too. So those are ovules. You don't see the other one behind that one when they took this section. So let's zoom in. So here are the ovules, three pretty decent images here. So that's an ovule, that's an ovule, and that's an ovule. So those ovules, that's where we're going to zoom in for eventually the egg to get produced. And so here's another image of the pine cone on the left has probably received pollen and is closed up. And then the pine cone on the right either is waiting to be pollinated, or in this particular case, they know that it's one that's releasing seeds because it's the second year. So let's look at that a little bit more closely. Let's assume, let's start from pollination. So pollination is a transfer of pollen to the ovule in the female cone. And then let's say that happens, and then the pine cone closes up, fuses up. Now 15 months later, the eggs will finally be ready. So the pollen is sitting there, and it will eventually have sperm produced in that pollen grain, but it's just sitting on the ovule waiting, waiting and waiting for the female part to do its job, which is to get that egg ready. All right, and then about 3 months, so the pollen lands on the ovule, and then it takes about 3 months for that pollen grain to germinate, which means it's going to form a pollen tube, and that pollen tube starts to extend down through the ovule tissue. So that pollen tube has to get through that initial front half of the ovule and make it to the egg. And so it's a job to get that to happen. And they often say, they use the term digest. It digests its way through the tissue to get to the egg. And then 15 months later, it finally gets there. So that's like what, a whole year to get that pollen tube to finally get to the egg. And then each pollen grain has 2 sperm. Well, okay, that was a mistake. 2 sperm discharge into ovule. Sorry, that should say ovule, not into egg. 2 sperm discharge into ovule. 1 sperm fertilizes the egg, the other sperm dies. And then a few months after that, you finally have the mature embryo inside the mature seed. So once you get fertilization, it's only 3 months that it has to develop to get to the baby, the baby embryo ready to go. So that's a long process. So roughly it is, most experts say with the gymnosperms, especially the Pinophyta phyla, it takes anywhere from 14 to 20 months from pollination to seed dispersal. So this is a big difference compared to most angiosperms. Most angiosperms are much quicker. Yes. Like tropical areas. As far as I know, yeah, I've never seen any, resources where they say, but on the other hand, tropical gymnos, have a totally, yeah. So yeah, it's a long process. So it's hopefully, if everything goes okay, though, they'll have those 2 seeds per scale that can get released. And then if those find a suitable habitat and start to germinate, grow, then they grow back. It's the sporophyte. It's really a baby sporophyte that's in that seed. And so now we're seeing some baby sporophytes here. one thing to point out is when conifers, especially when, needle trees, when they are growing at this young stage, they just have a single needle attached to the branches and stems. So if someone were to bring me something like this and say, what pine species is this? I'd be like, I don't know, because there's no defining clustering of the needles, and that's what you often have to use to identify these different pine species. So then eventually, a couple years into it, usually the new growth will start to shift into bundles or fascicles of the needles. And it can be, some still stay as one, but then they can have two or five or three. Just depends on the species, how they're going to cluster their needles into the fascicle. All right, and so their design is a little different. It's pretty thick hypodermis below the epidermis. It's just a little extra barrier of water, to protect against water loss in particular. And then they sink down their stomata, so they have sunken or recessed stomata, and they do that to slow down water loss from their body through transpiration. And then often you'll see resin ducts showing up because conifers produce usually a fair bit of resins and rosins. And then the odd thing about this is that you see an endodermis showing up in the pine needles. Endodermis is usually what we talk about in root tissue. So this is weird. And this is, as far as we know, these are the only leaves of any plant that has an endodermis around its vascular tissue. And I don't know, I've looked for like why. And I think in the lab write-up, I did mention one hypothesis is that they think it might have to do with preventing the more tender mesophyll cells here, the parenchyma cells, from freezing. So the ice crystals often form first in the vascular tissue area, and they think that endodermis is a barrier to stop the spread. Sounds plausible, but they still won't know exactly why there's an endodermis. It is kind of bizarre. And then inside the endodermis, that's where you have the vascular bundles. It's usually two like that, with the xylem and phloem clustered right next to each other. And then the rest of the pink area is just some parenchyma cells. So this is what it really looks like under the microscope. So the larger cells are the xylem, the larger red cells. And then the phloem is kind of sitting to the outside of that, more thin blue cells there. And then the sunken stomata. You can see a couple here on the left. Oh, there's sunken stomata there. And here. Other image, it's a little bit more noticeable. You can see sunken stomata. Several of them there. So again, just strategies to deal with the fact that these needles are long-lived. We say they're evergreen, but they do fall off. They just don't fall off the tree all at once. So they will have a certain percentage fall off every fall. But needles can be really long-lived, like decades they'll be on the tree. So they need to have these extra protections. Gymnosperm, all total in that group, about 720 species that we're aware of. Definitely the phylum Panophyta or Pinophyta is the biggest. Some people refer to this phylum as Coniferophyta. I'm going to probably stick with Pinophyta. That seems to be the more currently accepted term. So that's the conifers, and about 50 genera, and you can see 550 species. So by far the largest group. The genus Pinus, that genus, has about 90 species. And it is, so when you say that the Conifers are the most successful. What we mean by that is that there's more of them, more species of that group. That's how they often define success. Okay, but what about some of these other ones? So the cycads don't look like gymnosperms. In an ordinary person would look at that and probably say that kind of looks like a palm tree shrub. And it certainly looks like palm leaves. But this, believe it or not, is a gymnosperm. And there's about 11 different genera of cycads. So there's not that many out there, only 140 species. But they do have a true vascular cambium. They have true secondary xylem. They have the characteristics, many characteristics that they are confident to put them in with the gymnosperm. They are tropical and subtropical. That's where they're like, you don't find them here. It's too cold for them. And if you're a plant biologist, you would refer to when the dinosaurs were alive, you would say that it was the age of the cycads rather than the age of the dinosaurs, because cycads were so successful at that time point on Earth that they were all over the place and they were quite big as well. So that is claim to fame. Xanthiopula is the species that, as far as I know, is the only one native to the United States. It's found in Florida. And so in Florida, when I was there, I noticed people would put cycads as like living fences between their plots of the houses next to each other, which I thought was really quite pretty rather than a ordinary fence. But they aren't adapted to colder climates as you move north up from Florida. They don't like that. Now, if you go more tropical, you can find cycads that look like that, where they are as tall as trees. So, and in fact, most species are like that. The one in Florida is more shrub-like. This one, all the other species tend to be more tree-like and quite dramatic in their look. But again, you could see why someone would say, "Oh, that's a palm tree." It's easy to mistake that. One thing to be aware of, and they do sell cycads as potted plants for your house. Be aware that sometimes there's neurotoxins in the root tissue, so be really careful. And carcinogens, don't let your pets or kids chew on anything. And the leaves too. Don't forget about the leaves; those can have a neurotoxin as well. In the roots area, they often have cyanobacteria hanging out. And so that picture in the lower left is all the red dots. Those are cyanobacteria in the root tissue. And so these cyanobacteria are actually helping the plant get access to nitrogen. So these are bacteria that are fixing nitrogen. We talked a little bit about that before, how they can grab atmospheric nitrogen, N2, and convert that to ammonia. And then they'll feed that to the plant. So it's a nice win-win. The cyanobacteria get a nice home to live in, and they get some nutrients from the plant. They're really cool houseplants, but again, just be really careful that you don't have anyone eating any part of it. Okay, this is an even more bizarre. You would not, on the first glance, think that this is a gymnosperm. It's called the maidenhair tree. This is phylum Ginkgophyta. And ginkgo biloba is the only member that still has survived. Every other member of this phylum has died off. And very beautiful, broad, fan-shaped leaves. No needles here. And they can grow to be very tall. They're deciduous, which is again odd for gymnosperms. So they drop all their leaves all at once. And they're very popular for landscaping in city areas because they do look very pretty. The leaves are very majestic and they look really nice. Just be careful though. Oh yeah, and they're resistant to air pollution. The one thing to be careful though is that you want the male tree, not the female tree, because the female, when it's time for the ovules to get produced, and then the seed gets produced after fertilization, it gives off a really what we would describe as vile or rancid odor. And just by walking by the tree, you would smell it. So if you put that in the city park area, people aren't going to really want to go to the park because it smells so bad. However, if you can get past the taste or smell, apparently it's a delicacy. In certain areas of the world, people do consider it a delicacy. Kind of reminds me of lutefisk. Has anyone had the pleasure of trying lutefisk? If you're maybe have any Scandinavian relatives, my grandfather always insisted that my mom and my aunt cook up lutefisk for Christmas dinner. And it's white fish that's soaked in lye. You know, like the lye you use to make soap, and it's super caustic. So you soak it in lye and then boil it. Don't know who came up with that, but it's very smelly. It's so smelly that it's like you have to leave the house type smelly. It's nasty. But if you can make it past that and actually taste it, it's very mild. It doesn't have a super pungent taste. So this is another example. Super pungent odor, but not such a bad taste. So example of the tree, and then the female, which you don't want in your park. Oh, I mixed up the K and the G there. Ginkgo, it should be reversed. I tend to do that. Male cones on the left, pollen cones, female, the seeds here ready to be pretty much harvested. Okay, so that one you can see with the leaves, they're very fan shaped, very pretty, nothing needle-like. Okay, the last group of the gymnosperms is this phylum Nidophyta. The G is silent. Nidophyta, there's 3 genera that are still living: Nidum, ephedra, and Welwitschia. About 71 species. And so of all the phyla in this gymnosperm category, they are closely related to they're the most closely related to the angiosperms, because the reason they say that is they've, been dissecting these plants and looking at them really carefully and looking at how they go about their life cycle. And they have several features that are very angiosperm-like. For example, they have in their xylem, they actually have vessels. And I've been telling you over and over, only angiosperms have vessels. This is the noted exception. Okay. And it's a very minor exception, but it's notable because these are gymnosperms. So if you go back to the image further up in the slide set where we looked at the partial phylogenetic tree, you can see how the neophytes are the closest to the flowering plant offshoot here. So when it's drawn that way, what it means is that this group, the neophytes, are the most closely related to the angiosperms, more so than the other 3 groups here. Okay, so that's how you interpret that. So there's several characteristics. I'm not going to go into all of them, but probably the most notable is the idea that there's vessels in the xylem. That's really odd. Oh, I put the tree down here. There it is. So ephedra being shown on the left, that may ring a bell with you. And I'll talk about that. Needham in the middle, more of a vining plant. And then look at that weird Welwitschia. Looks like a hot mess, but it is successful in its particular area. So Needham is often just a vining plant that you see in tropical areas. And again, you'd look at that and you'd say, that's not a gymnosperm. That looks like an angiosperm, especially when you look at the female ovule. It's like, that doesn't look like a gymnosperm, but it is. Not around here. And the leaves are not needle-like. Ephedra, that may sound vaguely familiar. It's often referred to as Mormon tea. This is something that has been used for thousands of years, actually, in Asian medicine. It was used over here and a while ago, probably well before you were born, uh, for weight loss. There's a weight loss pill that you could take, and then people started dying from heart attacks. So they went, ooh, maybe we shouldn't do this anymore. So they had to pull it off the shelves. I think the FDA, no, the FDA didn't approve it. I don't think. But anyways, they pulled it off the shelf, and but it's still used in some medicines. It's just you have to be really careful. But they don't look like much. They just look like shrubs. There's no major leaves, so it's the stem tissue that does photosynthesis. You see these out west in drier desert conditions, the west and southwest. And then look at Welwitschia. Here on the lower left is a younger version. When it first starts to grow, it has two huge strappy leaves like that just come off kind of opposite each other. And then, believe it or not, all of this hot mess that you see on the top right, that is those two huge leaves. They get bigger and bigger and bigger, and then they shred with age, just through the damage caused by living a life where they don't shed these leaves. And so, very odd design. They are, as far as I know, the only place you can find them is Southwest Africa, and they're in the Namib Desert in that region, and they rely on incoming fog from the ocean as their water source. So incredibly well adapted. It's amazing that anything can survive, like a plant can survive in the Namib, but they do. And very, very unusual. And again, you'd look at that and say that's not a gymnosperm, but it is. Has some of the same features. So Welwitschia is the genus name. And there's really only one species left, Welwitschia mirabilis. So what can we do with gymnosperms? I can't get into all of it because the list is long. I'm going to be even this list is long, but it's longer still. If you start thinking about all the uses for gymnosperms, I'm sure you could come up with quite a bit that's already on this list. But this one you may not know about. This one is an amazing success story in terms of drug discovery. The yew tree, the scientific name is Taxus brevifolia. If you've ever heard Taxol, that drug, maybe you know someone who's taking that drug. And that was isolated from the bark of the yew tree. Well, Taxol is a billion-dollar industry for cancer treatment. It's a super effective drug for certain types of cancer. It can put people in total remission. So once the success of this drug was known, they started cutting down yew trees left and right and harvesting the bark. And then the plant scientists were like, whoa, you keep doing that and yew trees are going to extinction. So a couple of plant biologists said, well, let's go look at the yew tree more carefully. Maybe it's not just the yew tree producing this drug. Maybe it's the endophytes inside the tree. So the endophytes, I think I might have mentioned that maybe quite a while ago. Endophytes are the microbes that hang out inside the plant, just like we have millions of microbes that hang out in us. Similar situation. So they said maybe it's the microbe that's producing this drug. And so they went and isolated bark tissue and took it to the lab. Sure enough, a fungus that is hanging out in the stem tissue is producing Taxol. The yew tree can produce it too. So both the fungus and the tree can do it. But it's much easier to grow up the fungus for the drug than it is to harvest the tree and extract the drug. And so that's the beauty of endophytes. If you can find that the endophyte is doing the same thing that, you think this tree or this plant has this awesome chemical you want, it would be a good idea to say the second question should be, can one of its endophytes also do this? Because often it's easier to work with the bacteria or the fungi in terms of growing them up, especially to massive scales where you're trying to get a lot of that drug being produced. So it's been very successful for breast cancer, ovarian cancer, colon cancer, melanoma, and non-small cell lung cancer. Initially, it was for breast cancer, and now it's really they've shown that it can be very successful in several other types of cancers. So, billion-dollar drug, and a fungal endophyte is also there making it So that was a huge success story. And that was also a really good story to highlight endophytes and that they should not be ignored when it comes to applied uses of trees and plants in general. So, of course, when you think about pine trees, all the uses that we take advantage of here, the list goes on and on. Hemlock trees are known for certain, especially some red quality dyes, very beautiful dyes. The tannins that we use in various industries, they're usually harvested from hemlock. Certainly Christmas trees, obviously. Ephedra, again, that one has been heavily used for thousands of years. And if you do it right, it can be effective. Certainly essential oils, which is a big industry more and more these days. Ginkgo, I did it again. Keep reversing the K and the G. Um, so this has had a history, kind of ethnobotany history, that if you can take ginkgo biloba, they say it improves your memory. Have you heard that with ginkgo? Whether you take the supplement or they have teas. I did a little bit of reading into that. I was more interested in it for increased blood supply because I have Raynaud's syndrome where my toes and fingers go numb. My blood vessels constrict. They just spasm shut and then it looks like I have dead fingers. They just go white and very painful. And so I thought maybe ginkgo would help with blood supply flow. And I didn't notice a huge difference. Maybe I should try it again though. I only tried one brand. So as you know, non-FDA approved, so they don't regulate. You don't know what you're getting really. Anyways, ginkgo, some of the scientific studies that I read did show that it can increase the blood supply to the brain. And that's where people were making the jump that if it can do that, it probably helps your memory. That's a huge jump though. And so I don't think I would go that far. I haven't seen anything to make me think that way. But the data for increased blood supply, I think, is legit. And so, which is good. Having good blood supply to your brain is a good thing. That means you're getting lots of oxygen, keeps you alert, blah, blah, blah. So gin, again, probably $1 million industry to extract and create gin alcohol. Certainly burning of gymnosperms for fuel. And don't forget toilet paper. So, and the list goes on and on. All right, so gymnosperms, especially from phylum Panophyta, but some of the others too, have a long history and will continue. We make continued good use of them. Okay, so the next group is going to be the angiosperms. I guess we'll have to cover the part 2 of what I want to say about angiosperms on Monday. Are there any questions about the exam material or clarification on anything? As you're packing up, I'll stay a few minutes if someone does have a question you want to ask me.